So what was EMBL like when you arrived? In 78 was it?
79 in March.
Yeah.
It was just, everything was almost empty. There was almost nobody working here. I had space on the 6th floor there somewhere, and so first I did the only correct things. I tried to get oriented. I used to go to all the seminars of the biological groups. And of course I was sitting there and they were saying things which was a little bit Spanish village to me, but I knew, okay, after 3 months, somebody, it's like artificial intelligence. If somebody repeats so many times one word, then it's probably important, right? In order to enjoy it here and to be a valid member of that, that I had to somehow look for the collaboration with the biologists. Yeah. This was the really important thing, and in fact the biologists really cherished it as well, because usually some people came like physicists and they thought they were going to continue to physics no matter what, right? Applications there or there, maybe. Whereas I try to be developing something new, but something that people can use and will be lucky to use and happy. So that's where we started, and I first did some experiments only in my room there. I put some equipment there, and I was, in fact, the first year completely alone. And because I wanted to see what's kind of all the buffers mixing and all that, because this at the time was gels and these kind of things. And so then I went to some meeting that was organized here also by different groups, and there was also computer groups. And I looked at them and I said, guys, what are you sort of like doing? And they said what they were doing, they were computing the smiling effects and trying to straighten it up. And I said,
this is when you're running a gel.
Yeah, when you're running a DNA sequencing gel, and well, not only, but also protein, even the DNA fragments,
electrophoresis.
Gels, electrophoresis, right? And it was just sort of like smiling. And I remember that one night I forgot to turn on some equipment on it and some cooling, and in my system. And then I came next morning, I looked, I thought, my goodness, what happened? And so I thought about it, and I said, I guess it's because it's the temperature, but only indirect. The direct cause is the viscosity of the buffer, because that changes every degree 2%. So
you have a gradient.
Yeah, so you have you calculate easily the 20 degrees difference between the middle, where it's hottest, and between the ends, and then you calculate 20 times 2, about 40. So it just exactly correspondent to this. So I was happy with that. I understood what it was, and then I come to this meeting, to this conference where people are reporting heavily, how they try to develop software to correct it. And so I told them, guys, I might know how to do it. And they said, well, Wilhelm, you have been like 2 months, just continue a little bit there and then come back, and that maybe drove me to prove them that I knew it. So I went in and I met with, I took on this technology and all that. We met with Henrik Garoff, who was, of course, a very experienced molecular biologist already at the time, knew all this biochemistry of DNA sequencing, all the radioactive samples. And so I went to him and I said, if you would help me, I would help you to fix this, I will to do it. And he, at that time, everybody was cherishing his DNA samples because they were not easy to do it. The enzyme, everything was quite expensive. And then, so I said, but okay, but he had some samples which were old phosphorus, and they degraded very quickly. And he said, well, I can sacrifice those because in 3 weeks they will not be worth anything. So I was lucky, and we did something, in the system, and we put it on, and then because it was already quite old, so he had to put on his films and all these things with this cellophane on it, and first and the film, and then after about 2 weeks developing time, so he went and it was just a really nervous scene. We were about 5 people around, everybody crazy. Took it, opened, and it was very straight. So, okay, then he was happy that he did it. And then I immediately continued, in fact, with him, and already the gels were like, 0.5 millimeters at the time, even and that was a little bit too much, because when you put the film on it and cellophane, and then the film, then the radioactivity is spread, so the bands were like 2-3 times broader, therefore the resolution was much less. And so what I did, I washed out first all these buffers and all the urea. So we washed it first out. Then it was just pure polyacrylamide, and then we dried it down. And it was just only 5 microns or whatever, right? And then you put the film, so it was dry, thick, you could put the film directly without sulfide. Such nice bands. But the problem was that we first didn't know how to, the gels, when you separate the gels, they were floating everywhere. You lost it and everybody was unhappy. So what I thought, well, this is something I've used at Texas Instruments. It was a silanization of the service where you fix some grooves. So I had to find only the right silan. It was not so easy because then you had to make sure that not everything is kept and all that. But I found it, we tested it, and it was working like beautifully. You slip just the upper plate and the lower stayed there fixed.
And I can tell you, I've run hundreds of gels with silanized glass and peeling off and drying down the gels. I remember it well.
And in fact, this was so successful that afterwards somebody came from Sweden, from the LKB company at the time, and later Pharmacia, and they told me, that was really nice, we would be interested in producing. So I said, well, okay, so we made some patents, applications, and in fact this system was then produced at LKB, and Pharmacia later took it on, and in Amersham, in fact. So that was nice also for EMBL, and that's what I want to also emphasize, that at the time, in fact, we didn't do it sort of for personal glory. We were also happy that EMBL, which was a new lab, I was probably still trained from US, already from Texas Instruments. We were trained to be, whatever you do for the company. Whether at CERN, it was a little bit less, but also was, but particularly at EMBL, because it was new. So we always thought what you do, almost like John Kennedy said, don't think what you can do for but think what you can. So this was our approach at that time.
So, something, if you don't mind me saying, that always impressed me when I met you, because we overlapped for eight years at EMBL, and I have worked with a number of people with different physics backgrounds. Some physicists come to biology but are really interested in physics and really want to carry on their own project and apply it. Something that always impressed me about you and your approach to these sort of problems, I felt at least, was you actually engaged with the biologist who would be the users of the instruments or the equipment from the beginning and tried to design solutions that they could actually use.
That was exactly, you just hit it, and in fact it was from the fact that you don't want to just play in the corner. You just want to be useful and help to advance the field. And in fact, I was the happiest when somebody later on, after 10 years or so, said, if you have a problem and want to have it solved, go to Ansorge.
I think it's widely accepted that one of the biggest contributions to biology, or certainly one of the breakthroughs in biology that had the biggest impact, was genome sequencing and everything that's followed. And you obviously are famous for being someone who played a major role in making that possible by automating DNA sequencing and making it something that was fast enough and reduce the cost, so it became possible to scale DNA sequencing to a point. When I first sequenced DNA, Toby Gibson taught me how to sequence DNA when I was a PhD student. I would be there for 1,000 years if I was sequencing the whole human genome. So tell us the story how the human genome could be sequenced.
Yes, so this was the lucky coincidence. You have to be at the right time on the right spot. So one of the first invitation was at LMB to John Sulston and Aaron Klug, present in the room. Fortunately, I didn't know all these famous guys by name at the time, so I was in my way very relaxed. And when I finished, then I went to see John Sulston labs, and of course I met also Fred Sanger, tremendous impression on me. Then they told me, well, you're doing this DNA sequencing, but we are working now one step higher. They said, well, we are thinking about the human genome. That was at the time when when the discussions in 84, 85 in Science was going. And so we were trying to do now this human genome, and John Sulston was evidently from the beginning in it. And he was showing me how he took the outer radiograms and he was putting them through some scanners, but they were all smiling, you know, and they were still developing this software algorithms to trying to correct that and all these some things. So then fortunately came Brian Sprout to EMBL, and this was added sort of like next to my group, you know, by Philipson. He was so clever as well, such a good manager, you know. He knew this is what's the face what I call. I mean, you know, Kendrew and Philipson were the grounding excellence, bringing excellence to EMBL.
Yeah, and Brian also came from Cambridge, from the LMB.
Yes, exactly. So it was, you know, it's such a lucky, you know, I knew finally somebody who knows something so many times better than me, and I can therefore put it together. And so we're working on this, and he prepared the fluorescence compounds. So then the funny story is how do we get ahead, you know? So I couldn't ask Philipson at the time my training to give me 200,000 marks for laser just to test some idea, right? So I went to spectrophysics in Darmstadt, and I sat there and they were listening to me, and functionally again finding some very good guy who was open, and I said, well, you know, I would like this laser to borrow it just to see how it works, because we have some interesting things. And he asked me, so what are you trying to do? I said, well, we fluorescently labeled some biological molecules and want to see in separation in gels. So he immediately grabbed this one, and in 1 week we had the big, huge laser, really 3 meters long, you know, in the lab. And then we put in front of it about 10 centimeter gel, you know, with that one. And then the whole thing, how to construct it, in fact. So it preceded, you know, my training from CERN. You first take everything which you involved in the system. It has to be tested for the optimum properties. So it was like, for instance, the glass had to be the really cleanest glass possible in order not to put some fluorescent background to that, you know. Yeah. So this was one fact. Then we looked also at the quality of the buffers and all these kind of things. Everything has to be the purest possible. And then the minimum background towards the detectors. And so we realized that it had to be perpendicular to the polarization of the laser. And so then we put it on. We made some tests with it, Josef Stegemann, Christian Schwager, and me. And then we had this sample from Brian Sprout. We put it on. We looked at the electrophoresis. Bam! There was a peak from the laser to the, but because we had to make a special also how to feed the laser between the two glass plates. Yes. You know, that was the trick, because we put like 0.2, 0.3 millimeter thick gels only as well there, and it had to be optical quality and everything. So we had actually these coupling glasses, as we call them. They had to be prepared also by a special company and all this. And so it was really exciting. And then there was a huge laser, and there was a huge photomultiplier in front of it. And then it was the interesting. We put it in the dark chamber with all the systems set up, and with this huge laser and with this photomultiplier there. And I remember then came out. We just put only one track, you know, from the sequencing gel with this fluorescent, and then that finished about 11 in the night. I couldn't sleep anyway. I just had to go. So I took the film. Which we had before from the samples, and I took the recordings that Christian Schwager made from the computer systems to take the data analysis and data collection. And so I went to the huge lab, you know, there was a huge study lab opposite to Philipson, his office. And then I was putting it all on the table, and everything was big, huge. And I was standing there, and I had this radioactive film in front, and then was this paper peaks. And I looked, there is a peak here. Yes, there is a peak here, there's a peak. And when I was at the end, it was like about 140 bases. And then I realized somebody was sitting in the room next to me. It was about 1 hour after midnight, 1 o'clock after midnight, and it was Philipson sitting there. And he couldn't help it also.
So Leonard Philipson, really was director of EMBL.
Yeah, the director of the EMBL, and he was also trying to enjoy the moment of the first Eureka. Anyway, so it worked, and then we ordered.
But there's some really important. No, this is an amazing story of fundamental discovery, but there's some really important messages you see here, because it only happened because you had a multidisciplinary team first of all.
Exactly.
People with different expertise who would interact with each other. And second of all, what I'm saying is you're doing something where I could think of so many places it would be impossible for you to do this, because you would have to guarantee up front it would definitely work. And how could you know that? You would have to guarantee, you'd have to explain exactly how everything was going to work. How do you know that without trying it?
No, that was EMBL at the time. It was saying, we were all excited, and it was just the right place for all kind of advanced projects where you could not guarantee the results, but you put people, enthusiastic people.
And you trust them. And the director is passionate as well, and wants to give you the freedom and the opportunity to explore the possibilities. And this, of course, became a huge achievement for EMBL, that thanks to the way EMBL operated, you were able to build a group, collaborate, develop this technology. It must be so rewarding to see the impact of that work.
Well, it was great, and as you correctly said, I think it was possible only at the EMBL at the time, because we had the money, we didn't have the pressure, or you have to deliver tomorrow. In fact, the pressure was we. And so then was Christmas, I was in Geneva, and we had all these 4 multipliers there, and we getting that, well, the smallest that one could get, but still that you could get like 2-3 clones per gel only with them. And I was thinking, it was Noel, the 24th December, and then I was like, I stood up, I ran up, and I did that, and I wrote immediately email to Josef Stegemann. I said, Josef, immediately order diodes, the avalanche diodes systems, and try if you put it directly on the glass, because we have to have some optics to get it under the photometer. That took lots of space and all this. So I said, if you put it directly on the glass, and I couldn't sleep for 4 days. We couldn't, I couldn't sleep. Then I said, so what do you see? We called about 4 days later, and he said, it's just like the photomultipliers. Well, this was another fantastic thing. So then I knew we had a commercial system, when I was discussing in 1990 after the HPRT, when we then the system of EMBL was so performant that the
so the HPRT was an example of biological clinically important biological locus with Tom.
That was Philipson's contact with Tom Kasky.
With Tom Kasky, yes.
Tom Kasky, he from Houston Baylor College of Medicine. So he tried first these systems, and they realized that they didn't get the results as they would like to, and so then he met somewhere Philipson at some conference, and Philipson said, well, we have now some system. He was hoping, and so he came to me and said, now there's one guy from US, he would like to test it. I said, immediately, Leonard, you said we tried. So Tom Kasky came, sent 1 or 2 guys, we discussed, immediately started this very nice collaboration. And this was so fruitful, and it's always the combination of the technology with the biochemists and biologists.
And that demonstrates the impact.
Yeah, because this guy, he knew everything about HPRT, how it works and whatever, whereas it took me about 1 month to learn the epoxantin phosphoribosyltransferase transferase. So that was it, and this was gene locus. It was not only just the cDNA or something.
It was the whole thing.
It was the whole thing, and it was very important. And when we finished it, then Tom Kasky was even really also getting very high in the US because we then could correct all the other sequences which were published about it were not correct. Okay, there were like 5% errors or even more, he said. And with our systems, then he could get it very accurately, and he could then predict all the important things. He made even further developments, and Richard Gibson, in fact, is the guy who works with him, and he was then taken over this project. So they were all these things which were important afterwards for genetics, for determining paternity and all these things.
Yes.
They used it for. And the nice thing also was that because it was then commercial produced by LKB Pharmacia, later Emershem, that clinics were using it extensively, because they said this is, if I want to sleep well, the doctors, they said, I put samples on your system in 3 dilutions, and then I let it run, and I get some sequence. I see here is a mutation or not, and I can sign it and sleep well. So I think EMBL was, that was pretty, EMBL deserved to getting really reward from that. For us, we were proud when EMBL was getting, because we needed to push EMBL. Already during the time, and I'm getting to the important stuff, after the HPRT, I knew that if you want to do the human genome, like we published, what was the important thing about the HPRT was that then Jim Watson, when he wanted to really push the human genome then in America to get the budgets, he said if you want to sell it, you know, and in the Congress put it and show that it works, then you have to have this project, and then he concretely names the EMBL system that we have there developed, and that was used in Congress to justify the investment in it. So another really fantastic achievement for EMBL, I must say. You know,
yes, absolutely.
And this is a little bit less mentioned, in fact, because of course the American scientists, they love to be sure that some glory also comes to US, if not all, then most of it.
Well, it was a great advance for humans, for the world, but very important to recognize the major fundamental contribution of the EMBL. I mean, one of the other things I think you really played a fantastically important role at EMBL was setting up the core facilities.
Yes.
And so maybe you could just briefly describe what they were and how they impacted research at EMBL.
So it was, in fact, you know, when then, because we had with Kafatos then a little bit contact with European Union.
Yes.
So, in about
he had taken over from Philipson as the Director General of Kafatos.
93. There was 1 year was 93, 94 was actually acting John Toos, because Kafatos had still some obligations in Harvard. And then when he came, we started, in fact, I had already contact with European Union before. It was a little bit against the wish of Philipson. He was not enthusiastic. But then I saw him, well, you know, how do you prove the EU or the people around to do it correctly, if not the EMBL does it? So that was something he was sensitive to, so he allowed me to take grants, 1 or 2. And we were really well paid. I could employ like 3-4 people for that. It was excellent money, in fact, from that time for us, because with that technology it was easier for them, for the others. And so we decided that this technology now we could make it usable to many other people, also outside of EMBL. And then in about 1997, I put forward some proposal already, because we forgot to mention that at that time we had, since 92, we had also mass spectrometry.
Yeah, I was coming to that.
Yeah. So at that time, and it was also very adventurous, in fact, because, you know, I talked with Philipson like since 1990, and we said, you know, I said, you know, mass spectrometry is picking up in the US, and there are some really good guys, really interesting work doing. And he said, we need mass spectrometry. You know, it's just the way to do it. And he was the type of guy who could immediately understood, that's good, you know, let's do it.
I remember meeting you in the corridor and you telling me, I've just hired this young guy and he's going to revolutionize protein analysis. And I said, how are you going to do that? And then he moved Matthias Mann into the lab next to mine, and we had our offices back to back.
That's it.
And this was one of the biggest impacts on my future career.
Yeah. Well, you see, and you have to also be ready to overcome some obstacles, because, you know, not at that time, as you remember, not every biologist was happy to spend money on some more instrumentation or things around, right? They thought every money should be going to the biology. And then I thought, well, okay, then we have to get somebody who is a rising star, you know, somebody who is still modest, just finished PhD or something. But so screening a little bit around, going to some conferences, and then it was this Matthias Mann. And so I spoke with him, and I realized that's going to be the guy, because he was not only intelligent and clever and looked like what he would like to do, but I could see he would not get lost here, okay? Because it was easy to. I mean,
it was a great choice.
Yeah. And so I was so really happy. So then we had a fantastic department, Brian Sprout, Matthias Mann, and to our team for the we had DNA sequencing, plus the cellular biology, imaging, and microinjection, all these things. And And then Matthias Mann, and then he took the Matthias films also, you know, he took over. And of course they were immediately running like as fast as they could, probably even faster, you know, and combining also the DNA sequencing with the proteins. It was just another fun, you know.
It's super exciting, and I think the biologists caught on pretty quickly just how useful this would be.
I then remember seeing Kai Siemens and Eric Arcenti going out from the lab, you know, also like the hair like that, and thinking what else can they do. So, okay, another satisfaction of something. You had to take the risk, right? It's not. I mean, can you imagine if the mass spectrometry would only consume money and no results, you know?
But really, to make big advances, you have to invest resources, and you have to give people time and trust them and give them an effort to do things. And I think you've just given us a fantastic series of examples of what the rewards can be.
Well, and we were still the generation that, you know, we tried to spend not too much money, but we went step by step always. First you prove feasibility at this step, that can encourage you to do the second step, which may be a little bit more costly, but still limited in the amount. And then when you know it's working, then you can do the big stuff, right? And this is something that I'm not so sure that everybody does nowadays. You know, we knew EMBL was sort of like financial not so bad. At least all the universities around us were envious of the money we had. But it was not an endless budget, right?
No, the budget absolutely is not endless, never was, and it's important that you emphasize that money was not unlimited, and that was a factor. You clearly have played a really major role in delivering impact through EMBL, helping to realize the vision of the European countries who established this transnational organization. And now, as you, I'm sure, are aware, we're in the 50th anniversary year of the founding of the EMBL organization. As we come to the end of our conversation, could you just say a few words about what EMBL means to you?
EMBL gave me, in fact, my career, so I should say, with all the preparation before. But whenever I did here in the life, if I achieved something, that the main part of it is definitely of EMBL. So I can encourage everybody, if you have the chance, come to EMBL, and in every state, enjoy the support of the EMBL and the collaboration here.